In the previous video, I talked about the structural classes of neurons, the bipolar, unipolar, and multipolar. Here we have three functional classes of neurons: sensory neurons, motor neurons, and connectors. Connectors that are located in the spinal cord called interneurons. We've got a lot of those and lots of sensory neurons. Our sensory neurons are unipolar. The cell bodies are located in what are called ganglia, like clumps of cell bodies. And they are going to deliver information, senses, from our receptors to the CNS, and that's incoming information, so it's afferent. And again, here's a review of the information it's bringing in. Well, information from external receptors. An external receptor detects change in the external environment, like touch or temperature or pain. And proprioceptors, that's a new one. Proprioceptors are found in our joints and muscles and kind of monitor where our body is, the position and movement of our bodies. And then viscera means organs. So the receptors that monitor our internal organs. This incoming information from the sensory neurons comes to the spinal cord where the interneurons live. And these interneurons process the information. They do some planning, some learning in the CNS. Now, the motor neurons carry information from the CNS out to do some action. The action, those organs that act are called effectors. And the effectors are muscles and glands. That outgoing information is efferent, efferent fibers. So there's two types of outgoing information. Some that is voluntary, that would be somatic motor neurons. And some that is involuntary, that would be, that would be like the visceral motor neurons, the automatic stuff we have no control over. And that would be the skeletal and smooth muscle and the glands. So you might want to pause here to look at this graphic to understand the neurons. What type of neuroglia is found in the PNS? Oh yeah, it's the only one you have to learn. Schwann cell. Which of the following is not one of the functions of astrocytes? Astrocytes do not produce myelin. Which of the following is in contact with cerebrospinal fluid? Well, you don't need to know this one. What's in contact with it are the cells that make it called the ependymal cells. This is a completely different topic. We saw this a little bit in the muscle chapter. We saw that membranes can have what's called a membrane potential. That is, they can have a charge difference. And it's because the fluids are different inside and out with different electrolytes and different charges. So the membrane potential is an unequal distribution of this charge where the inside of the cell is negative and the outside positive. These are very, very, very slight differences. The membrane is going to keep these differences because there are ions and pumps that move those ions back and forth across the membrane. So here we have a cell. The plasma membrane is right here in the center. Inside is slightly negative. Outside is slightly positive compared to inside. So outside we've got some sodium and some chloride, and inside we've got potassium and proteins. So here's kind of an important little topic. It's almost like our cells are bathed in salt water. So outside the cell is the salt. Look at that. NaCl is salt. outside the cell, extracellular fluid, is the salt water. But the cell doesn't want that. The cell wants to have lots of potassium inside. So, what actually happens is the sodium kind of leaks in all the time. Some sodium and potassium channels are always open. Sodium leak channels, so the sodium is leaking in. But there's not a whole lot of potassium outside, so the cell has to grab the potassium and pull it in. So it does so with this active pump, and we saw this in chapter 3, the sodium-potassium exchange pump. It is this sodium-potassium exchange pump that keeps the membrane potential accurate. What it does is it pumps out 3 sodium for every 2 potassium it pumps in. Remember, sodium keeps leaking into the cell, so it's got to pump it out. It wants more potassium in the cell, so it's got to pump it in. There are some leak channels that just keep happening, and then here's this active, requiring ATP, sodium-potassium pump. So when the membrane potential changes, as we saw in the muscle chapter, it can cause an action potential in the membrane. And it can cause an action potential in the nervous system. And it can cause our glands to secrete. When the cell is resting and there's a membrane potential set up, but the cell's not doing anything, it's not transferring any information, it is called a resting potential. And if there's a stimulus, there's a slight switch in the ions. A slight change, and this is called the graded potential. Well, if the stimulus is big enough that it causes a big enough graded potential, it's going to trigger a nerve impulse or action potential. Let me say that again. A resting cell membrane potential is called a resting potential. The cell is not transferring any kind of information. But then there's a stimulus. And if the stimulus causes a slight switching of the ions, and this is called a graded potential. It might not do anything. It might not go any further than that. But if this graded potential is big enough, then it will stimulate an action potential. So the action potential will travel down the axon to the axon terminals. And inside those axon terminals, just like at the neuromuscular junction, that presynaptic cell has neurotransmitters, and they will be released. They will cross the synaptic cleft, bind to receptors on the postsynaptic cell, and change the membrane on the second cell. But here's the thing. It's not just one axon connecting with the second axon. Thousands of axon terminals can communicate with the neuron cell body. So let me see if they have, where's that picture? They don't have a picture here. So here we have one cell connecting with one cell. But imagine, instead of just these axon terminals here, there are thousands of them coming in to this neuron here. That's the way our nervous system is. It's not one-on-one. It's thousands on one. So basically every single neuron has those neurotransmitters. Well, a neurotransmitter has 2 choices. It's either a stop neurotransmitter or a go. So if it's a go, the neurotransmitter will stimulate the action potential. If it's a stop, the neurotransmitter won't do anything. So there's some kind of processing going on this second cell. This second cell, if it has thousands of different neurotransmitters coming into it, has to process that information and add them all up and decide whether to go or to stop. This is our simplest form of information processing. Does the signal go on or does the signal stop? Which of the following occurs next after an action potential reaches an axon terminal? Does the neurotransmitter diffuse across? Does calcium trigger exocytosis of the neurotransmitter? Well, we don't talk about calcium, but they're just saying, is the neurotransmitter released? Does the neurotransmitter trigger the opening of ion channels on the second cell, or does the neurotransmitter bind to their receptors? Well, the neurotransmitter first has to be released. And then A will happen. It will diffuse across the synaptic cleft. And then D will happen. It will bind to the receptor. And then C will happen. The neurotransmitter will trigger the opening of ion channels. And when ions enter that second cell, it can cause an action potential. So we'll look at the phases of an action potential. So remember that the cell is set up with positive charges outside, negative charges inside. It's polarized. Well, once an action potential reaches this cell, it's going to cause a depolarization of the membrane. That is, the neurotransmitter, say like acetylcholine, opens the sodium channels on the second cell. The acetylcholine is released from cell number 1, the presynaptic neuron. It's going to cross the cleft and open the sodium channels on the postsynaptic neuron. And once the sodium enters the cell, it causes a change in the ions. That is, if positive charges are rushing in, it's going to make the inside less negative. And that's why on a graph like this, here you have, this would be positive up here, negative down here. The sodium rushing in is going to cause less, to become less negative on the inside. It's polarized. Then the sodium channels close, and instead of sodium, this is a little weird, instead of sodium rushing out to go back to normal, because remember, sodium rushed in, changes the charges, but instead of sodium rushing out, actually potassium rushes out. So you can imagine the cell is not returned to normal. It's lost a lot of potassium. It's got too much sodium in there. So we know that sodium-potassium pump is going to work like mad to get the membrane back to normal so another action potential can take place. So let's go over this again. A graded potential, that is, there's been a stimulus and there has been a graded potential, which is a change in the ions across the membrane. If the change is big enough, then there's going to be an action potential. The action potential is going to travel to the end of the axon, and the neurotransmitter is going to be released. It's going to cross the cleft and be bound and open sodium channels, causing a larger depolarization. The sodium channels close, potassium channels open, and potassium leaves the cell, and the cell is repolarized. But then the sodium-potassium pump has to reset everything back to normal.